Good afternoon, folks. I hope you're all having a good day. This is the beginning of my last lesson for anatomy and physiology concerning the human reproductive system, or as I like to put it, how to make an entirely new human in only 9 months with simple tools you probably have around the house. If you are in my anatomy and physiology course, you should probably go on the Blackboard site and download a copy of my handouts so that you'll have reference to vocabulary terms and a summary of key concepts. I also strongly recommend that you go into your textbook or your lab manual and follow along with some diagrams of the reproductive system so you can see what I'm talking about while we do this, because if I hold up those pictures here, YouTube will probably take my video down. All right, so as you are probably aware, human beings, like all animals, engage in sexual reproduction. And if you don't know that, well, you're in for a wild ride with what we're about to talk about here. What that means, essentially, is that all of our offspring contain equal genetic information from both of their parents. You provide half of the genetic data for your offspring, that your partner provides the other half. You might recall that humans have exactly 46 chromosomes in the normal karyotype for our species. 23 of those will come from mom, 23 will come from dad. To put it another way, we are what biologists like to call diploid or 2N organisms, D-I-P-L-O-I-D. Any organism that is diploid has paired chromosomes. Now that means in order for us to form as a diploid organism, we can't reproduce with diploid cells. If half the data is coming from mom and half the data is coming from dad, we have to reproduce using specialized haploid, H-A-P-L-O-I-D, or 1N cells, which have only one copy of each chromosome. That is 23 chromosomes each. The two haploid cells combine to make one diploid offspring. Those cells, by the way, are known as gametes, G-A-M-E-T-E-S. That is the general term for sex cells. They are synthesized in organs called the gonads, G-O-N-A-D-S, which are the gamete-producing sexual organs. The male gamete is the spermatozoa, S-P-E-R-M-A-T-O-Z-O-A, or sperm for short. The female gamete is the oocyte, O-O-C-Y-T-E. That's two O's at the beginning. Note that you pronounce each of them separately. It is oocyte, not oocytes. Sometimes you'll see two little dots over the second O to indicate these are both pronounced separately from each other, but you don't have to write it that way. In any case, though, the normative way that sexual reproduction works is that the oocyte is a large, non-motile cell produced by the female of the species, and the spermatozoa are small, highly motile cells produced by the male of the species. That in sexual reproduction, the small motile spermatozoa travels to and penetrates the large non-motile oocytes. That allows them to combine their genetic information, 23 chromosomes from dad, 23 chromosomes from mom, in order to produce a single fertilized egg known as a zygote, Z-Y-G-O-T-E. We all begin as a zygote, a single cell formed from the fusion of egg and sperm, in order to make one singular diploid unit, which will then begin undergoing a cycle of division, turning from a zygote into an increasingly complex ball of cells called an embryo, and then eventually from an embryo into a fetus, and then eventually from a fetus into a functioning human being. Now, we are not going to go through all the different stages of embryonic development in here. That's a little beyond the scope of this course. If you take my Biology 2 class with me, you will learn more about that. If you are watching on this channel and you want to go on a lecture I have for another class, you can look for my biology lecture on Animal Fundamentals Part 1, which goes into the details of animal development and all the stages of development from zygote on to functioning organism. But for this course, we're not really going to go into that in detail. What I'm concerning myself with right now, what we're going to be discussing for the next couple of videos, is going to be the details of the human reproductive system. That is the set of organs, structures, and responses that are responsible for our ability to produce and nourish offspring. So the reproductive system is concerned with a number of things, not only with delivering sperm to eggs, but also with facilitating the subsequent development of the offspring into a functioning infant, as well as providing food for that infant in the earliest stages of its growth, and as well as regulating many aspects of anatomy, development, and behavior in both the male and the female of our species. You're going to find that our reproductive systems are going to be responsible for a lot of the changes that happen in our bodies at puberty and a lot of our structural and functional developments, and indeed a great deal of our behavior. For those of you who have not taken me for an evolution class, it is worth pointing out, and this is a good time to mention this as we go into this particular lesson, that your DNA chiefly and primarily is concerned with being copied. That the fundamental function of DNA as a molecule is to make itself copied, and that is to get itself into new cells. Reproduction for any living organism is fundamentally a mechanism for creating new vehicles, new cells, that can contain and nourish our DNA. The evolutionary biologist Richard Dawkins wrote a book called The Selfish Gene about this, basically offering the premise that the fundamental feature of evolution, contrary to what you might think, people say survival of the fittest, Darwin himself would have disagreed with that contention, that it's not survival, it's reproduction, that it's really all about getting your DNA into the next generation. The key point of evolution is not who survives, it is who reproduces the most. Because if you reproduce, then the features that helped you to reproduce become more common in subsequent generations, and your DNA is being propagated through your offspring. It doesn't matter how well you survive, evolutionarily speaking, if you don't reproduce, all the adaptations that helped you to be good at surviving die with you. So fundamentally, the point of evolution is reproduction, and what this basically means is that most aspects of animal development, one way or another, directly or indirectly, are shaped around reproduction. That really your body, viewed at its most basic, fundamental, stripped-down evolutionary level, is a fancy tool for copying your DNA. And that means that the reproductive system of all your bodily systems is the one most concerned with developing and shaping the rest of you in a way that will make you better at doing that thing. Any of you who want to know more about the biology of sex and reproduction in this way, I also have another lesson on this channel for my Biology 2 class, a biology lecture entitled Negative Frequency Dependent Selection and Sexual Selection, which deals with the way in which sex has shaped evolution. So you might want to check that out if you are interested further in this topic, or just contact me directly and I'll be more than happy to either talk with you more about it, the evolutionary side of things, or point you to resources related to it. For the time being, we are going to concern ourselves primarily with the end result of that evolutionary process, the specific anatomical, behavioral, and physiological features that our species possesses, which give it the ability to reproduce, and how that affects the rest of your body. Now, there are some things that the human reproductive system has in common across both sexes. Ultimately, the reproductive system is responsible for producing sexual hormones, which control various aspects of our sexual developments, our anatomical development, and our reproductive behavior. And our reproductive systems are both responsible for the production of gametes. Gametes are, of course, produced in the gonads, the reproductive organs. They are produced from a population of cells called germline cells, Germ-line, from a process that you probably remember from your Bio 1 class known as meiosis, M-E-I-O-S-I-S. During the process of meiosis, the germline cells will undergo a 2-round cycle of division to become gametes, that is, eggs or sperm. Now, those things are the same across both male and female reproductive systems, but beyond that, they are going to diverge greatly. The reproductive system is very interesting, not only in humans, but across the animal kingdom, in that it is the only significant bodily system in which there are meaningful structural and functional differences between male and female organisms. That there are some small things across the board that we can expect to see that are a little bit different in men versus women. For example, when we talked about the urinary system, we noted that men have an internal urethral sphincter that women do not have. So there are little differences like that that we can expect to encounter in other bodily structures. But this is the only bodily system in which we have a wholesale different organizational pattern for the male of our species as opposed to the female of our species. Now, I do want to point out two things about this before we get underway. First off, you will note my usage of the word sex rather than the word gender. Do not use those words synonymously. They do not mean the same thing. Sex is a biological construct. Sex is a way of describing two different reproductive models within a sexually reproducing species, with the male being defined essentially as the sperm producing and the female being defined as the egg producing. That the sperm is the projective, if you will, gametes, that the egg is the receptive gametes, that the male reproductive system is organized around delivery of sperm to the female reproductive system is organized around receipt of sperm from the male and subsequent development of the young, whether that means into an egg that the female lays or into an offspring that develops in mom's body until she gives live birth, like in humans. So when we define sex biologically, that's what we're talking about, two distinct reproductive models. Now, I should point out that in the course of human sexual developments, sex is also not a binary. In no species that has sexes is sex a binary. There is a lot of ambiguity. There are a lot of middle ground areas. It is best perhaps to think of sex as a bimodal curve with two sharp peaks with what we would think of as anatomically male and female on either end, but in between a much less common but nonetheless existent spectrum of variation in which you can have some degree of expression of male and or female anatomical features and physiology in the same individual. This is not as unusual as you might think, but thinking of sex as a strict binary ignores a tremendous amount of important variation that exists in our species and indeed in all sexually reproducing species. For purposes of this lesson, we are going to be focusing on the, you might say, classic male and female anatomical and physiological structures. Most of the features involving ambiguous or mixed genitalia are features that are not reproductively viable in any case, and therefore they're somewhat outside the scope of the discussion we're about to be having here. But it's an important thing to keep in mind that exists. Now, gender. I'm not going to be using the word gender throughout this discussion, and you shouldn't be either, because gender and sex are not the same thing. Sex is a set of biological features explaining a reproductive and developmental model for an organism. Gender is a social construct. It is a way of understanding the rights, roles, privileges, and perception of an individual in their culture or their society. Gender is in most cultures, but not all cultures, mapped to anatomy, as in that you have male genders and female genders, but it is not a perfect correlation. We in the Western world are somewhat unusual in that our default cultural assumption is that gender and sex are the same thing and that there are only one of each, or the two of each. There's one sex that is the same as one gender and one sex that is the same as the other gender. In point of fact, this is a very unusual perspective for human beings as a whole. The overwhelming majority of human societies recognize the existence of more than two genders, including some genders that are not necessarily mapped to one set of sexual features. So I will not be using the term gender in here. I will be using the term sex because gender is a sociological and anthropological term discussing a role of an individual in society, not a set of anatomical characteristics. So if you want to know more about gender, I strongly recommend take a cultural anthropology class. Really, everybody should do that anyway. Cultural anthropology teaches you how to be a better human, but it's not going to be what we're talking about in here. All right, so that being said, we have to kind of start by defining what our parts are and what they are going to be doing. So first off here, let's kind of just lay out the basics, the fundamental division of male and female reproductive systems. I'm going to begin with the male simply because the male reproductive system, quite frankly, is the simpler of the two. The male reproductive system is a lot less anatomically and physiologically complex than the female reproductive system because, quite frankly, it has an easier job. The male reproductive system is mostly about delivering gametes to the female, and once that's been done, the male has no further role in the reproductive act. By contrast, the female reproductive system not only has to receive gametes from the male, but then subsequently develop and nourish the growing offspring. So the female reproductive system of the two is definitively the more complex one, and we'll be spending more time on it. But I feel like talking about the male first is important, A, because it's shorter, and B, because most of the important things that happen in reproduction happen after the sperm have been delivered. Therefore, getting that part out of the way first then sets us up for talking about what happens in the female body subsequently. But let's lay out the basic parts and functions of each before we go any further. Male reproductive system in humans is going to consist of a few distinct parts. We have the testes, T-E-S-T-E-S, also known as the testicle, so that's a more colloquial term. We have the penis, P-E-N-I-S. We have the scrotum, S-C-R-O-T-U-M, S-C-R-O-T-U-M. And we have a number of associated ducts and glands, such as the seminiferous tubules and the prostate gland. Functions of the male reproductive system, first off, the production of sperm in the testes. 2, the sustenance and nourishment of those sperm to complete their development. Thirdly, the delivery of those sperm by way of the penis to the female reproductive tract. And fourthly, regulation of male growth, development, and reproductive behavior, mostly facilitated by the production of male sex hormones, most chiefly testosterone. T-E-S-T-O-S-T-E-R-O-N-E, testosterone. Now, the female reproductive system is going to have more parts, more complexity, and more things that it does. Female reproductive system is going to consist of the ovaries, O-V-A-R-I-E-S, singular is ovary, O-V-A-R-Y. The uterine tubes, U-T-E-R-I-N-E, tubes, also called the fallopian tubes, capital F-A-L-L-O-P-I-U-S, I-P-I-A-N, sorry, my bad. I was thinking the name of the guy who discovered them. The uterus, U-T-E-R-U-S. The vagina, V-A-G-I-N-A. The vulva, V-U-L-V-A. The mammary glands, M-A-M-M-A-R-Y. And a variety of other supportive glands and structures. Female reproductive system has several distinct roles, some of which are analogous to those of the male. First off, the female reproductive system is responsible for the production, storage, and nourishment of oocytes or eggs. Secondly, for ovulation, that is the release of eggs on a periodic basis, O-V-U-L-A-T-I-O-N. Thirdly, reception of sperm from the male and transfer of that sperm to the location of the oocytes for fertilization. Fourthly, nourishment and protection of the fetus as it develops. Fifthly, nourishment of the offspring following birth. Sixthly, actually we should put this one first, I suppose, parturition. P-A-R-T-U-R-I-T-I-O-N. Parturition is a fancy word for childbirth, or that is the delivery of the infant once its development is complete. And finally, of course, the female reproductive system also is involved in the production of female sex hormones, chiefly estrogen, E-S-T-R-O-G-E-N, and progesterone, P-R-O-G-E-S-T-E-R-O-N-E, which are responsible for regulating various aspects of female reproduction, behavior, and anatomical development. So you can see the female reproductive system has a lot more distinct things going on than the male. All right, that's kind of giving us our basic layout of the parts and jobs of the various reproductive structures in the male and the female of our species. So let's begin by going a little bit more in detail into the male reproductive system. Anatomically speaking, the male reproductive system can be divided into two basic components. We have the external male genitalia, G-E-N-T-A-L-I-A, genitalia, or genitals in the singular, which the male external genitalia are the penis and the scrotum, that is the sac that surrounds the testes. You might have thought, okay, aren't the testes external genitalia because they're located outside the body? Nope, the testes are internal genitalia because they're inside of that sac. And then we have, of course, the internal male genitalia, which includes the testes, several different ducts, such as the epididymis, E-P-I-D-I-D-Y-M-I-S, the ductus deferens, D-U-C-T-U-S, deferens, D-E-F-E-R-E-N-S, and a number of glands, such as the seminal vesicles, S-E-M-I-N-A-L, vesicles, the prostate gland, P-R-O-S-T-A-T-E, and the bulbourethral glands, B-U-L-B-O-U-R-E-T-H-R-A-L. We can also count the urethra as one of the ducts present in the male reproductive tract. It is a duct that, of course, fulfills a double role, being part of the urinary tract as well. This is a uniquely male feature of the reproductive system. The female urethra is not a reproductive structure, and the male urethra is. Okay, so let's deal with the external genitalia first. So we'll start with the scrotum. The scrotum, S-C-R-O-T-U-M, is a sac-like structure which contains the testes. The scrotum is divided into distinct left and right compartments, separated by a partial septum between them. Mostly, the scrotum consists of a layer of loose skin overlying a layer of loose connective tissue, areolar tissue, and that in turn overlying a layer of smooth muscle. There are actually two distinct smooth muscles associated with the scrotum. Most of the scrotal sac is comprised of the dartos muscle, D-A-R-T-O-S. And there is also a suspensory muscle located in the abdomen called the cremaster, C-R-E-M-A-S-T-E-R. Those two muscles control the movement of the scrotum. And you might think, okay, well, does the scrotum actually move? Yes, in fact, it does. The most important movements of the scrotum, involving both the dartos and the cremaster, involve drawing the testes closer to the body. Now, there are two reasons why this would happen. Well, three reasons. One, the testes draw closer to the body immediately prior to ejaculation, immediately prior to orgasm in the male. The testes tighten and pull closer to the abdomen. Secondly, this is done to protect the testes, that whenever there is danger in the environment, the testes, being in an externally suspended sac in the body, are going to be a vulnerable point, as any male who has ever been bopped in them can firmly attest. Now, during times of danger, say you're in a fight or flight response, the testes will often be drawn up closer to the abdomen. By having them more closely held against the abdomen, it makes them less likely to be damaged in the course of conflict. There is a reason that the withdrawal of the testes is associated with a fear response. Now, anybody who possesses a set of testes hearing this is no doubt wondering, as they have certainly wondered at some painful moment in their life, why the heck are these very vulnerable organs located outside of my body? And the reason for that is getting to our third reason why we have muscles to control the movements of the scrotum, and that is to regulate the temperature of the testes. Because here's the thing, folks. The testes have as one of their chief jobs the manufacture of sperm. We'll talk more about that in a few minutes. What you may not realize, however, is that testes require a different operational temperature than the core human body temperature. You probably recall from other classes, core human body temperature, textbook average is approximately 37 degrees Celsius, give or take about, half a degree on either side. That's about the normative range of variation. But right around 37 degrees Celsius is where you spend most of your time. The enzymes that manufacture sperm require an operational temperature of approximately 35 degrees Celsius. So about 2 degrees Celsius, or almost 4 degrees Celsius cooler than core body, almost 4 degrees Fahrenheit cooler, I should say, than core internal human body temperature. As a result of this, if the testes were located up inside the body cavity, the warmth of the body core would render them incapable of making sperm. And this gets down to the entire reason why the testes are located in a sac that is suspended further away from the abdomen, because that means they're not surrounded by the core mass of the body, and therefore it is easier for them to maintain a lower internal temperature that facilitates the production of sperm. You will find that in warmer conditions, the cremaster and dartos muscles relax more, causing the testes to hang further from the body. You will find that in colder environments, those muscles tighten in order to draw the testes closer to the body and help them to maintain the temperature that is required. So that's the third function of those smooth muscles associated with the scrotum, is to regulate temperature in the testes. I should point out an interesting thing about this, that you are going to find that the scrotum as a structure is very flexible. It has a lot more space in it than you might think is needed. That in normal conditions, the scrotal sac is fairly loose. The testes feel fairly mobile within it. You may not realize it, but they're actually fairly tightly held in place by connective tissue. It's just that the skin they are attached to is very loose, which gives the impression that they can shift around quite a lot. However, also keep in mind that under normal circumstances, the muscles associated with the walls of the scrotum are in a relaxed state. They only tend to tighten under the very limited conditions that I mentioned, and therefore that gives the testes a certain amount of mobility within the scrotal sac while, normal conditions prevail. All right, actually, I started talking about the external genitalia, but while we're in the scrotum, let's pause for a moment and talk about its contents. Let's talk about the testes. And by the way, spelling consideration here, testes is the plural form, T-E-S-T-E-S, not I-E-S-E-S. If you're talking about just one of these organs, it is the testis, T-E-S-T-I-S. So one testis or two testes. The term testicles is a colloquialism, but it is anatomically, we say testes or testis more commonly. This is also one of those cases where the word origin is not necessarily corresponding nicely to a Latin or Greek root that is descriptive of the body part itself. Actually, I kind of like the origin of this one. You know that we try to give structures names that correspond to, things about them that tell you something about where they are or what they're doing or whatever. So, we can talk about, for example, the bulbourethral gland, which is associated with the bulb of the penis and also with the urethra. So that's a very anatomically descriptive term. The word testes or the word testicle comes from a Latin root, and it does not mean anything having to do with the anatomy. It has a little bit more of a culturally oriented meaning. The word testicles, translated literally from the Latin, means little witnesses. So that's where we get the origin of testicle, and from there, testes or testis. All right, so the testes are paired organs in humans. That's the normal condition for almost all male animals. The testes are small ovoid organs, about the shape of grapes, located inside of the scrotum. Average male testis is between 4 and 5 centimeters in length. And they are interesting structures in that they have both endocrine and exocrine functions. Their endocrine function, of course, involves the production of male sex hormones, that is testosterone. We'll talk more about that a little bit further down the line. The exocrine function, of course, is going to involve the production of semen, or at least the spermatic component of semen. The rest of semen is produced by other glands and structures. So production of sperm is an exocrine function of the testes. Now, when you're looking at each testis, you will find that within the scrotal sac, each testis is surrounded by an outer capsule of dense, irregular connective tissue called the tunica albuginea. Tunica, Tunica, albuginea, albuginea. That surrounds the entire testis on the outside. Ingrowths of that reach into the interior of the testis and divide it up into a number of internal compartments called lobules, L-O-B-U-L-E-S. Average testis has between 300 and 400 lobules separated from one another by septa that are formed from ingrowths of the tunica albuginea. Each of those lobules contains a tightly coiled seminiferous tubule, S-E-M-I-N-I-F-E-R-O-U-S, tubule, T-U-B-U-L-E. So each lobule contains a seminiferous tubule, very tightly wound, coiled tube. Between all of the lobules, each testis contains about a half mile of seminiferous tubules. The seminiferous tubules are going to be the site of spermatogenesis, S-P-E-R-M-A-T-O-G-E-N-E-S-I-S, spermatogenesis, or the production of spermatozoa. The seminiferous tubules are going to be where we are going to make the sperm. Each one is surrounded by a layer of cells known as interstitial cells, I-N-T-E-R-S-T-I-T-I-A-L, interstitial, cells between the spaces. Interstitial means between spaces. Interstitial cells are also called cells of Leydig, capital L-E-Y-D-I-G, named for the physician who first described them. Cells of Leydig are the ones that secrete testosterone. They're the same thing as interstitial cells. Now, all of these seminiferous tubules are going to empty into small ducts called tubuli recti, T-U-B-U-L-I, tubuli, recti, R-E-C-T-I. If you're talking about one of them, it is a tubulus rectus. The tubuli recti, in turn, converge on a singular duct known as the rete testis, rete, R-E-T-E, testis, T-E-S-T-I-S, which carries sperm out of the testis. Now, an important detail about testis development. Although the testes reside in the scrotum in the developed male, they form inside the abdominal cavity. The testes and the ovaries, the female gonads, are equivalent organs. And if I didn't say this before, the testes are the male gonads. They are the male reproductive organs that produce hormones and gametes. Testes are male gonads, ovaries are female gonads. They are both analogous paired structures. They both develop internally within the abdomen. However, in the course of male development, the normative thing that's going to happen is that the testes are going to descend into the scrotum from the abdominal cavity during the course of development. It is not entirely unusual for a male baby to be born with undescended testes. That condition is called cryptorchidism. Cryptorchidism. C-R-Y-P-T-O-R-C-H-I-D-I-S-M. Cryptorchidism. From crypts for hidden or deep, orchidism from orchid. Cryptorchidism literally translates, I love the poetry of the language here, as hidden flowers. So cryptorchidism is the condition of undescended testes, in which they're still in the abdominal cavity at birth. It is normally self-correcting. A male born with cryptorchidism will normally experience descent of the testes within 6 months of birth. However, if the testes fail to descend, that will result in that individual being sterile. That if undescended testes will not be able to produce sperm properly simply because they are in too warm of an environment to do so unless they're in the scrotum. All right, so what do the testes do? Chief function of them, of course, is spermatogenesis, the production of spermatocytes, or sperm, as we call them. Now, this is interesting because this is a thing that you do not do your entire life. Spermatogenesis does not begin in the male of our species until puberty. That is, males do not make sperm at all until they hit puberty. One of the changes that happens in the male body with the onset of puberty is the beginning of spermatogenesis, triggered by stimulus from the follicle-stimulating hormones produced by the pituitary gland. At puberty, the male will experience the growth and proliferation of interstitial cells, therefore producing larger amounts of testosterone, as well as enlargement of the seminiferous tubules and consequently enlargement of the testes as a whole. Now, the seminiferous tubules are where the action happens. This is where sperm get produced. You will find that within the seminiferous tubules, we are going to have spermatogonia, S-P-E-R-M-A-T-O-G-O-N-I-A, spermatogonia, which are the male germline cells. And if you think back to your Bio 1, the germline cells are the cells that undergo meiosis to make gametes. Now, spermatogonia are going to undergo both kinds of division. They are among the very few cells in the body that experience both mitosis and meiosis. They undergo mitosis to make more spermatogonia. You got to keep replenishing them. And then some of them, however, are going to undergo meiosis in order to make the gametes themselves. So a spermatogonium, whenever it undergoes meiosis, is going to first transform into a primary spermatocyte. This is the beginning stage of its sperm development, that the spermatogonium transforms into a primary spermatocyte. S-P-E-R-M-A-T-O-C-Y-T-E-S. Primary spermatocytes are tetraploid cells, or 4N cells, T-E-T-R-A-P-L-O-I-D. Tetraploid means having 4 copies of each chromosome. In other words, they have doubled their chromosome count prior to division, so temporarily in preparation for sperm production, your spermatogonium is going to turn into a primary spermatocyte with 4 copies of each chromosome for a grand total of 92 chromosomes in humans. Now, that primary spermatocyte, and it's S-P-E-R-M-A-T-O-C-Y-T-E-S, spermatocyte, that primary spermatocyte will undergo meiosis. After meiosis 1, it will split into 2 diploid cells with 46 chromosomes each, called secondary spermatocytes. From there, each of those cells will undergo meiosis 2. You might recall meiosis has 2 rounds of division. At the end of meiosis 2, each of those will then split into 2 haploid, or 1N cells, with 23 chromosomes each, 1 copy of each chromosome. Those haploid cells that we get at the end of meiosis 2 are called spermatids. S-P-E-R-M-A-T-I-D-S, spermatids. So that's the end result of meiosis. We're going to have 4 spermatids, each of which has 23 chromosomes, 1 copy of each chromosome. In other words, exactly half of a human chromosomal set. The spermatids, in turn, are going to finish development into spermatozoa. Little fun fact I would like to put in here is remember that biological sex is determined by the 23rd chromosome primarily. That the 23rd chromosome comes in 2 varieties, the X chromosome, which is much larger and more complex, and the Y chromosome, which is a very small chromosome in humans. In fact, the Y chromosome only has about 77 functional genes in it. The Y chromosome is a very, very small chromosome. Its entire job is to regulate the details of the development of male anatomy, male reproductive features and processes. That's what Y chromosome controls. The default sex for all sexually reproducing organisms is female. Male is a modified female. It is the presence of the Y chromosome and the genes on it that facilitate the development of male structure and function from the base female structure and function. We know this because if you have a human female that is born with only one singular X chromosome, a condition we call Turner syndrome, by the way, that we will see external female presenting anatomy, external female presenting genitalia, even though that individual will be sterile and will never undergo normal puberty. So that is the default anatomical structure. In any case, however, the chromosomes, sex chromosome division-wise, the 23rd chromosome of the 23 in humans is the so-called sex chromosome. In female humans, there are 2 copies of the X chromosome. Each cell in the body of a chromosomally female human will be XX for the 23rd chromosome, 2 copies of the X chromosome. In the male of our species, each cell for a chromosomally male individual will be XY, 1 X chromosome, 1 Y chromosome. Everybody gets an X chromosome from mom. You need an X chromosome for survival because there are genes on the X chromosome that do things that everybody requires. The X chromosome is not just involved in reproductive structure. It contains more than 1,100 genes. You cannot survive without an X chromosome. Some of you might be aware, for example, that genes that control such things as blood clotting and the development of color vision are found on the X chromosome, which is why X-linked chromosomal disorders are responsible for things like color blindness and hemophilia. So everybody gets an X chromosome from mom. When mom's gonads undergo meiosis, each of them is going to have an X chromosome as its 23rd chromosome. When dad's germline cells undergo meiosis, the X and Y chromosomes are going to split apart from one another. In other words, any given sperm will either get an X or it will get a Y, get one of the 2. So whenever a male makes sperm, exactly half of the sperm he makes will be sperm with an X chromosome, exactly half of them will be sperm with a Y chromosome. And that is what determines the biological sex of the offspring. You've all heard, I'm sure, lots of old wives' tales and things about what you can do to facilitate getting a male child versus a female child, like certain times in your reproductive cycle in which, male children are more likely, or certain foods you can eat to make a better environment for the conception of males versus females. No. In mammals, that is not how it works. If you're a reptile, there are outside conditions like temperature that can affect whether the offspring turn out male or female. But in a male mammal, including ourselves, it's all determined by which chromosome is found in the sperm. And there's nothing externally you can do that makes it more likely that an X-bearing sperm or a Y-bearing sperm will get to the egg first. The male produces exactly equal numbers of each. It is completely a coin flip which one happens to make it to the egg first. And I'm sorry, no matter how much you want a male or a female child, there's literally nothing you can do that will make it more or less likely for any given sperm to be the first one to get to the egg, and whichever one wins, that determines the sex of the offspring. So it is always dad, incidentally, therefore, that determines the sex in sexually reproducing species, or in sexually reproducing mammals, I should say. It is a tragic detail of our history that in patriarchal societies, which have often valued male offspring more than female offspring, women have for centuries been blamed for a failure to produce sons, when in point of fact it was technically dad's fault the whole time. I mean, not that dad had any more control over it than mom, but mom's body was not the one making that determination. All right, but I digress. So we have undergone our basic steps of spermatogenesis here. We started off with a spermatogonium. The spermatogonia then become tetraploid primary spermatocytes. During meiosis 1, those divide into diploid secondary spermatocytes. And during meiosis 2, those divide into haploid spermatids. Those spermatids will then go on to finish their development into complete sperm or spermatozoa. Now, talking a little bit about sperm anatomy here. If you're looking at a sperm, you all know the basic shape of a sperm. The sperm has three basic components. It has a head. The head is the main cellular mass of the sperm. It's where we find the nucleus. It's where we find the chromosomes and the organelles. And on the head of the sperm, at its most anterior portion, you're going to find a mass of proteins called the acrosome, A-C-R-O-S-O-M-E, which literally translates as upper units or upper mass. The acrosome is a collection of enzymes clinging to the head of the sperm, forming a sort of cap on the head of the sperm. The acrosome is needed in order to do two things. One, to penetrate the oocyte. The enzymes on the acrosome are going to allow the sperm to break through the membrane of the egg and deliver its chromosomes. The other thing that those acrosomes do is that the acrosomes literally digest their way through the female reproductive tract to get to the egg in the first place. Keep in mind, and we're going to talk about this more later, that the female reproductive tract contains a lot of mucus. It has to, because that's a significant protection against outside bacteria that might get in. Since the vaginal orifice is an opening to the outside of the body, it provides an entry point for pathogens, and therefore the female vagina and the female uterus and so forth produce a lot of mucus to trap pathogens that have penetrated, and then they can be discharged. The sperm have to get through exactly that mucus. They have to get through that same protection. And the enzymes in the acrosome not only help the sperm to penetrate the egg, but to digest a pathway through the mucus of the female reproductive tract so that they in turn do not get trapped by it and they can actually get to the egg. Indeed, most sperm are going to be exhausted before they even get there. But we'll talk more about that later. So we've got the head, which has the acrosome on it. This is going to, serve to get us to the egg in the first place and help us penetrate it. We have the midpiece, which is a small segment located posterior to the head. The midpiece is going to be the power source for the sperm. The midpiece contains the mitochondria of the sperm, and it generates energy to power the sperm's motility. The midpiece contains a lot of mitochondria relative to its size, because sperm are the only kind of cell produced by the human body that needs to move under its own power. The only cell that's responsible for its own individual locomotion without being propelled by the bloodstream or something else. And therefore, the sperm have to have higher ATP production than any other cells to power their motility. Important feature about that. The midpiece does not, under normal conditions, penetrate the egg during fertilization. As a result, although chromosomal DNA is delivered into the egg, mitochondrial DNA from the sperm generally is not. So in terms of your chromosomal DNA, you're exactly half mom and half dad. You've got 23 chromosomes from your mother, 23 chromosomes from your father. But all of your mitochondria, under normal circumstances, came from mom. That the mitochondrial DNA is exclusively found in mitochondria, but because the midpiece of the sperm, where the mitochondria are, is not going to be penetrating the egg, that means that dad's mitochondria do not get into the egg. All of your mitochondrial DNA, therefore, came from your mother's mitochondria, from the mitochondria present in the oocyte. And therefore, genetically speaking, you're a tiny little bit more mom than you are dad, because your mitochondrial DNA, barring novel mutations, is identical to that of your mother. We have very recently learned, as in the past year, that there are very, very limited occasions when a few mitochondria might actually get into the egg from the midpiece, in which case there is a very slight chance that you might have a small amount of mitochondrial DNA from your father, from those of his mitochondria that penetrated the egg. But that seems to be vanishingly rare. We do not even know how rare it is yet. No more common than 1 in every 200,000 fertilizations, if not less than that. But it does seem that once in a very, very, very great while, some of the mitochondria from the midpiece do get into the egg during fertilization. Finally, we have the tail of the sperm. The tail extends from the midpiece. The tail is largely a flagellum, a long whip-like fiber of the motion protein actin, which whips back and forth to propel the sperm forward. Now, completing spermatid development into spermatozoa. The spermatid, when it first forms, does not have all these features. It does not have an acrosome. It does not have a tail. Those features are added to it as it finishes its development in the seminiferous tubules. In the walls of the seminiferous tubules, you're going to find a population of cells called sustentacular cells. S-U-S-T-E-N-T-A-C-U-L-A-R. Sustentacular. They are also called Sertoli cells, capital S-E-R-T-O-L-I, after the guy who discovered them, or sometimes called nurse cells. I don't care which name you use, sustentacular cells or nurse cells are what you hear more often, but if you hear them called Sertoli cells, I'm not going to complain about that. Whatever you call them, however, your sustentacular cells, you might have noticed in there the root word sustenance, to sustain. The sustentacular cells provide nourishment. They provide energy for the developing sperm. They also produce a number of hormones involved in development of the sperm, including androgen hormones, A-N-D-R-O-G-E-N-S, or masculinizing hormones. Estrogen is produced in the sustentacular cells. You might not realize this, but the male body does make a tiny amount of estrogen. The female body does make a tiny amount of testosterone. Most male estrogen is produced by the sustentacular cells. And there's a group of hormones called inhibins, I-N-H-I-B-I-N-S, which regulate the development of the cells here. They inhibit overdevelopment. The nurse cells also form a blood-testis barrier. Just like you have a blood-brain barrier that segregates your bloodstream from the brain and limits what gets into the brain, we have a blood-testis barrier that lies between the spermatogonia and the complete sperm. The entire point of that blood-testis barrier is it shields the sperm from your immune system. Basically, it protects the sperm from being exposed to white blood cells, which might misidentify them as a pathogen and destroy them. A breakdown of the blood-testis barrier will result in sterility because your white blood cells will attack your sperm as if they were invaders. Now, the sperm complete their development in the walls of the seminiferous tubules, being nourished by the sustentacular cells. They develop their acrosome, they develop their tail. And once their development is complete, they are released into the lumen of the seminiferous tubule. From there, the sperm leave the seminiferous tubules and are collected by the tubuli recti that I mentioned before. All of those tubuli recti converge on a single vessel called the rete testis. And the rete testis is going to move out of the testis, and going to connect via another tube, the efferent ductule, E-F-F-E-R-E-N-T, efferent ductule, D-U-C-T-U-L-E, to the epididymis, which is the next stage in our process. So our pathway of movement here is sperm form in the walls of the seminiferous tubules. They travel seminiferous tubule to tubuli recti, to rete testis, to efferent ductule, and then finally to the epididymis, which is the next stage in our process. Epididymis, that's spelled E-P-I-D-I-D-Y-M-I-S. Be careful about, people always want to reverse one of the I's and the Y's. E-P-I-D-I-D-Y-M-I-S. Now, the epididymis is a cone-shaped structure located on the posterior and inferior surface of each testis. The epididymis has three basic parts. It has a head. The head contains the end of the efferent ductule. It has a body. The body contains a multi-meter coiled tube called the duct of the epididymis, and then finally a tail, which contains an efferent duct that leads away from the epididymis. So what's going to happen is that sperm will enter the epididymis. They will move from the efferent ductule into the body of the epididymis, into the duct of the epididymis. They will remain in the epididymis for 12 to 16 days. During that period, they are going to finish their developments. They are going to complete the growth of their acrosome, and they are going to complete the development of their tail. In other words, they're going to gain the ability to move and the ability to fertilize. That is finished in the epididymis after being begun in the testes. So sperm are going to move through the epididymis, entering via the efferent ductule, passing through the duct of the epididymis, and exiting via the tail of the epididymis. From here, the sperm move into a tube called the ductus deferens. Ductus, D-U-C-T-U-S, deferens, D-E-F-E-R-E-N-S. Sometimes it is called the vas deferens. Vas, V-A-S, you might remember that term, means vessel. The vas deferens is a tube approximately 45 centimeters long. It emerges from the tail of the epididymis. That tube is going to ascend along the posterior surface of the testis, kind of climb up the back of the testis, and then it is going to join other structures to form the spermatic cord. S-P-E-R-M-T-I-C, the spermatic cord, C-O-R-D. The spermatic cord is a bundle of separate structures that are all kind of grouped together. The ductus deferens is part of it. The testicular artery, T-E-S-T-I-C-U-L-A-R, the testicular artery that supplies blood to the testes, is part of it. The testicular vein, which drains blood from the testes, is part of it. And so is the testicular nerve, which provides sensation to the testes. So ductus deferens, testicular artery, testicular nerve, testicular vein, all collectively form the spermatic cord. That spermatic cord is surrounded by two layers of protective connective tissue, the external and internal spermatic fascia, F-A-S-C-I-A. The spermatic cord is also surrounded by the cremaster muscle, which you might remember mentioned before. Spermatic cord leaves the scrotum. It extends through the abdominal wall, entering the abdomen through a passageway called the inguinal canal, I-N-G-U-I-N-A-L. Think back to your basic anatomical regional terminology. The term inguinal means pertaining to the groin, and the groin is the juncture of thigh and abdomen. The spermatic cord passes right through that region. The inguinal canal, I should point out, is potentially subject to being blocked by an inguinal hernia. Some of you who are athletes might experience an inguinal hernia, or you might know of an athlete who was put on the disabled list for a while because of one. An inguinal hernia, H-E-R-N-I-A, bulges into the inguinal canal and could put pressure on the spermatic cord. That can lead to extreme pain, as well as blockage of blood supply to the testes and blockage of sperm leaving the testes. Inguinal hernias are normally surgically correctable. Now, the spermatic cord exits the scrotum via the inguinal canal. It extends immediately, I should say, across the pelvic cavity, moves behind the urinary bladder, passes through the prostate gland along the way, P-R-O-S-T-A-T-E. We'll talk about that in a few moments. The prostate gland is going to surround that tube. You're going to find that smooth muscles in the walls of this ductus deferens are going to propel the sperm along via peristalsis. They are mostly moved by muscular action. rather than via their own flagella, because that conserves their energy for use later. So mostly your body is moving the sperm by itself along that pathway. Now at the distal end of the vas deferens, it's going to enlarge, it's going to open up into a larger space called the ampulla of the ductus deferens, A-M-P-U-L-L-A. You probably figured out by now when you hear about an ampulla in anatomy, that's an enlarged end or reservoir. Now, adjacent to the ampulla of the ductus deferens, you're going to find the seminal vesicles, S-E-M-I-N-A-L, vesicles, V-E-S-I-C-L-E. The seminal vesicles are going to be joined to the ampulla by a tiny little tube about 2.5 centimeters long called the ejaculatory duct, E-J-A-C-U-L-A-T-O-R-Y. The ejaculatory duct then projects into, oh, sorry, I should say, okay, all right, so back up a second here. So we got seminal vesicles, we've got ampulla of ductus deferens. Those converge to form the ejaculatory duct. So both the seminal vesicle and the ampulla of the ductus deferens are going to empty into a common passageway, which is the ejaculatory duct. Ejaculatory duct projects into the prostate gland and ends at the urethra. So we have a passageway here that sperm can follow there to go out of the ductus deferens into the ejaculatory duct, and then finally to enter the urethra, which of course is going to be their passageway out of the male body. Now, of course, the passageway out of the male body, we're going to flip back to our external male genitalia for a moment here and talk about the male penis, P-E-N-I-S. Now, the penis is the external organ of copulation, C-O-P-U-L-A-T-I-O-N. Copulation is the term for the physical act of sex, the insertion of the penis into the vagina, also called intromission, I-N-T-R-O-M-I-S-S-I-O-N. Some texts, older texts might refer to the penis as the human intromittent organ, I-N-T-R-O-M-I-T-T-E-N-T. An intromittent organ is a penis or equivalent structure in an animal for the delivery of sperm into the female reproductive tract. Not all animals with a male reproductive system necessarily have a penis as such. There are a couple of other intromittent organs that are used in other kinds of animals, but for humans and for mammals in general, the penis is what we're going to be dealing with here. Now, the penis, in terms of its internal structure, look inside the penis, you will find 3 distinct columns of what we call erectile tissue, E-R-E-C-T-I-L-E. 2 of those are called the corpora cavernosa, C-O-R-P-O-R-A, corpora, cavernosa, C-A-V-E-R-N-O-S-A, or cavernous bodies. The corpora cavernosa form the dorsal and lateral portions of the penis. The third erectile column is going to be the corpus spongiosum, corpus C-O-R-P-U-S, spongiosum, S-P-O-N-G-I-O-S-U-M. The corpus spongiosum forms the ventral portion of the penis. It surrounds the urethra. The urethra passes through the corpus spongiosum. At the distal end of the penis, the corpus spongiosum expands to form the glans penis, G-L-A-N-S, or just the glans for short. That's a singular word, even though it has an S at the end. The glans penis is the flared tip of the penis. The glans penis is at birth protected by an external sheath of loose skin, technically called the prepuce, P-R-E-P-U-C-E, more commonly referred to as the foreskin, F-O-R-E-S-K-I-N. Now, interesting thing about this, that here in the Western world, it is a very common cultural practice rooted deeply in Abrahamic faith religious traditions, at birth or shortly after birth, to cut off the foreskin of the male, to surgically remove the prepuce. That is an operation called circumcision, C-I-R-C-U-M-C-I-S-I-O-N. circumcision. In Western society, circumcision is relatively common, particularly in the United States. It is a very common cultural tradition here. Indeed, male infants are circumcised kind of by default unless the parents say, no, don't do that. Now, there's a lot of argument about whether or not this is a good idea. Again, it's a common cultural practice. In some religious traditions, it's considered a very important practice, particularly in Judaism, in which the circumcision is an important ritual of rite of passage for the developing infants. Medically speaking, the removal of the prepuce or foreskin is completely unnecessary. It is not a thing that has to happen for any medical reason. There is nothing that is going to be functionally different in a male with an intact foreskin. The foreskin forms a sheath that covers and protects the gland's penis. It pulls back during sex, exposing the glands. Now, an intact foreskin serves as a protective tissue around the glands, increases its nervous sensitivity and its sensation, and protects it from damage. There is some evidence suggesting that an intact foreskin creates a pocket around the glands in which pathogens can accumulate. And there are some studies suggesting that uncircumcised males are more prone to STIs and to infections around the tip of the penis associated with this. There's some circumstantial evidence that penile cancer is more likely in males who have an intact foreskin. And males who have an intact foreskin are also potentially subject to yeast infections, which can accumulate in the gap between the glands and the foreskin. However, all of these are problems that are remedied with good hygiene. And increasingly, doctors are saying, okay, let's not circumcise by default. Let's not cut a piece off of a non-consenting male baby. At birth, there's no reason to remove, medically speaking, and basically saying, hey, if you want to get a circumcision later in life, you can electively do that, but we're not going to mutilate this child without their say-so, prior to a time when they're able to think about it and consent to that operation. So there are some measurable medical benefits to circumcision. There are some measurable medical drawbacks to circumcision. It's a cultural debate right now. All right. But I will tell you again, at a biological level, no need for it. No reason why the prepuce needs to be removed. All right, now, in terms of the structure here, we are going to find that the proximal end of the penis, often called the root of the penis, is formed by an expanded mass of tissue called the bulb of the penis, B-U-L-B. The bulb of the penis is going to be formed from the proximal end of the corpus spongiosum. It extends to form the crus of the penis, C-R-U-S. The crus of the penis is made out of the corpora cavernosa. So the bulb of the penis is formed from the corpus spongiosum. The crus of the penis is formed from the corpora cavernosa. The corpora cavernosa, the crus of the penis, anchor the penis to the pelvic bones. And so these structures root the penis in place. Externally, the penis is surrounded by a layer of loose skin, which is highly elastic, able to stretch during erection. That skin becomes very thin over the glans penis, which is responsible for its more pink coloration. You're seeing blood vessels through it. The glans penis is highly innervated. It is actually the most innervated structure in the male body, a high amount of sensation, lots and lots of receptors there, and the thinner skin makes them more sensitive. Now, one of the chief functions of all of these erectile columns is that in preparation for the sex act, they will become engorged with blood. They will fill up with blood, and causes them to swell and expand, forming an erection, E-R-E-C-T-I-O-N. Erection is both a process and a structure. We can talk about the process of erection, by which the penis lengthens and hardens, or we can talk about an erection, which is a term for an erect penis. That firming and lengthening of the penis provides mechanical leverage or mechanical advantage by which it can be inserted into the vagina without collapsing. that making it a hard, rigid structure enables it to be smoothly inserted, deeply inserted, and to move in a rhythmic fashion. You're all wondering, so I will answer the question for you. In terms of average dimensions throughout our species, and this is the species-wide average, the average flaccid penis, F-L-A-C-C-I-D, that is the resting length, if you will, the non-erect length. The average flaccid male penis is about 8.9 centimeters long, or around 3.5 inches. The average erect male penis is approximately 12.9 to 15 centimeters in length, or in other words, between 5.1 and 5.9 inches. That's about the normative range. Basically, between 5 and 6 inches is the normal species. When I say normal, I say the average species length for an erect penis. Average circumference when erect is approximately 12.6 centimeters, or about 5 inches. Obviously, there is considerable variation in individuals. Couple of centimeters either way is not at all unusual, but anatomically speaking, as long as it's a functional organ, the length is not significantly a factor. There is evidence suggesting that a longer penis is more likely to result in fertilization, for the simple reason that sperm are being introduced more deeply into the female reproductive tract. But in a lot of cases, that doesn't even matter, simply because even a longer penis is often stopped by the cervix whenever it is penetrated. All right. Now, there are a couple of accessory glands associated with the male reproductive tract that I kind of glossed over here. So finishing up our look at the internal male genitalia, let's talk about the accessory glands. First off, I mentioned way back when we talked about the ampulla of the ductus deferens that it is surrounded by the seminal vesicles. The seminal vesicles are small sac-like glands. Each is about 5 centimeters long. They're located next to the ampulla of the ductus deferens. The seminal vesicles produce a thick mucus-like secretion that is high in sucrose, that is your basic disaccharide sugar, also high in a variety of other nutrients, vitamins, minerals, amino acids, and so forth. A variety of chemicals called post-ejaculatory clotting agents, P-O-S-T-E-G-A-L, pardon me, P-O-S-T-E-G-A-C-U-L-A-T-O-R-Y, got lost in my spelling bee in my head there. My post-ejaculatory clotting agents are going to be responsible for causing the semen to clump together in the acid environment of the female vagina, which protects it from too much exposure to the acidic secretions therein. So that fluid, the fluid produced by the seminal vesicles, makes up about 65% of the volume of semen, S-E-M-E-N. Semen is the fluid that is ejected from the penis during ejaculation or during the sex act. Semen, about 65% of it is secretions from the seminal vesicles, mostly post-ejaculatory clotting agents, nutrients, and sugars. Now, also, we have the prostate gland. Please be careful of the spelling. Somebody always says prostrate gland, P-R-O-S-T-A-T-E, prostate. The word prostrate means to lie on your belly. The prostate gland is about 4 centimeters long, 2 centimeters wide. It surrounds the proximal end of the urethra and the base of the ejaculatory ducts. So both the urethra and the ejaculatory ducts are going to pass through the prostate gland. It's located right at the base of the urinary bladder. Now, the prostate gland secretes a thin, milky fluid called prostatic fluid, P-R-O-S-T-A-T-I-C. Prostatic fluid is released directly into the urethra. It makes up about 30% of the volume of semen. Prostatic fluid is released into the urethra via between 15 and 30 prostatic ducts. The number varies from one individual to another. The purpose of the prostatic fluid is primarily to neutralize acids encountered by the sperm. Partly that is neutralizing acids found in the urethra itself. Urine is normally slightly acidic, you might recall, and we want to neutralize that acid before the sperm enters. The prostatic fluid is also, of course, projected into the female reproductive tract as part of semen, and it serves to help neutralize the acid environment of the vagina. Sperm require a pH of between 6 and 6.5 to operate. They like a slightly acid environment, but the acidity of the vagina is normally about 3.5 to 4. So by having that prostatic fluid there, it's a basic fluid that buffers that environment, raises the pH slightly to make the environment more hospitable to the sperm so they are not destroyed. Prostate gland also produces a substance called PSI, or prostate-specific antigen. PSI serves to thin the semen, making it more liquid so that it flows more easily. Now, PSI is also interesting because prostate-specific antigen will rise in its count in the bloodstream during prostatic cancer. So an elevated level of PSI is an early warning sign of prostate cancer. Now, finally, in terms of our accessory glands, the last one we have the bulbourethral glands, B-O-L-B-O-U-R-E-T-H-A-L. The bulbourethral glands are tiny pea-sized glands, about the shape and size of peas. They are located near the membranous parts of the urethra, M-E-M-B-R-A-N-O-U-S. The membranous part of the urethra, if you remember from our anatomy of the urinary system, is the proximal end of the urethra, and you will find the bulbourethral glands around there. They get their name because they are near the bulb of the penis and around the urethra, bulbourethral. The bulbourethral glands produce, mucus. The mucus makes up about 1% of the volume of the semen. So in terms of overall composition, if you have not been paying attention, or let me just summarize for you, even if you were, 65% of the volume of semen is secretions from the seminal vesicles, sugars, nutrients, and post-ejaculatory clotting agents. 30% is secretion from the prostate gland, prostatic fluid, and PSI, which neutralizes the acid environment and thins the semen. About 1% of the volume of semen is mucus produced by the bulbourethral glands. And that means that that's about 96%. Only about 4% of the volume of semen is actually sperm. So using the words semen and sperm synonymously is actually not accurate at all, because only about 1/25 of the volume of semen is actually sperm cells. Now, semen, of course, is the fluid that will be ejected from the penis during ejaculation. Semen is going to be produced continuously by the male body from puberty onward and throughout life. Normally, the sperm counts per milliliter of semen. So every milliliter of semen contains, on average, in a healthy adult male, anywhere between 75 million and 400 million sperm. That is the average contents of sperm, the sperm count, if you will, for the average healthy adult male, 75 million to 400 million sperm per milliliter of semen. Normally, an ejaculating male during ejaculation, E-G-A-C-U-L-A-T-I-O-N, ejaculation, that is the projection of semen from the penis. Normally, ejaculation releases 2 to 5 milliliters of semen at a time. That's about the normal volume of the average healthy male's ejaculates. A little bit higher, a little bit lower, but especially if the male has ejaculated recently, the volume will be lower. If the male has not ejaculated for a while, the volume will tend to be higher. So what that means, if you do the math on here, is that during the average ejaculation, a male human can be expected to release anywhere between 150 million and 2 billion sperm. Massive, massive, massive numbers of sperm. That is necessary because most sperm are never going to get anywhere close to the egg. We say, reproductively speaking, in evolutionary terms, we say that male animals are the quote-unquote fast sex, which means that males produce lots of gametes very quickly. It is easy for the male body to replenish gametes every single week. The average healthy adult male makes enough gametes to personally repopulate the entire world. More gametes than there are people on Earth every single week. On average, healthy adult males can make between 7 and 8 billion sperm per week. So we're producing large numbers of them, but they are small, stripped-down cells without many features. They basically have what they need to get to the egg and deliver their chromosomes. From puberty onward, males continue to make sperm throughout life. Sperm count will gradually decline with age. It typically peaks in the mid-20s. The early to mid-20s is usually when male sperm count peaks. It will slowly decline thereafter, but males can remain fertile for their entire adult life. Now, once those sperm are introduced into the environment of the female reproductive tract, they've got a job ahead of them because they have to find their way to the egg, which means they have to pass through the vagina, they have to pass through the uterus, and they have to enter the uterine tubes, which is where fertilization will actually occur. Now, they are presented with a lot of obstacles in the course of doing this. Lots of things have to happen here to make this go. They have to, first off, get through the defenses of the vagina. The acid environment of the vagina is inherently hostile to incoming cells. It has to be to protect the female body from pathogens. And the sperm are no exception, which is why they rely on prostatic fluid to de-acidify the environment, to neutralize it and make it safer for them to move through. They have to penetrate the thick mucus that lines the female reproductive tract, also as a defense against bacteria. And that means they have to basically digest their way through. The acrosome on the head of the sperm is responsible for that. So the sperm are making their way forward deeper into the female reproductive tract. They are digesting their way through the mucus along the way. Inevitably, some of them don't make it. Some of them get destroyed by the acid environment. Some of them are going to get caught in the mucus. Some of them are going to use up their acrosomes getting through the mucus and then either become trapped in the mucus, die, or if they get to the egg, not have anything that can fertilize it left. Furthermore, sperm can get lost. They can swarm all over the place. They can zoom around in the uterus and the vagina. They're dudes. They're not asking directions. And so they are going every which way. Some of them will try to fertilize the inside walls of the uterus, which they can't do. Some of them will just get lost along en route and never make their way anywhere close to the egg. So basically what you're going to find is that the first sperm out the gates, the fastest ones that initially get into the female reproductive tract, they are the ones that are going to die first. They're the ones that are going to have the least likelihood of actually making it all the way to the egg, because they're going to get destroyed by or trapped by the hostile environment of the uterus and the vagina. The ones behind them are going to make it. And this is why we have to have lots and lots and lots of them, because the overwhelming majority of sperm will die, become trapped, or get lost before they get anywhere near the egg. Releasing hundreds of millions of them at a time gives us the possibility that a few of them will make it to the egg, because basically the first ones through are clearing the way for the ones behind them, digesting a path, and more or less sacrificing themselves so that the next ones in line have a shot at getting there. So just keep in mind, if you're worried about not coming in first with something, if you don't like that you didn't place well in the race or you didn't get the highest score in the class, remember, coming in first is not always the best thing. You were the result of the slowest sperm. All right, so that is going to be our pathway of the sperm and going to be how it ends up making its way from the male testes into the female reproductive tract. Now, we do have a little bit more we have to say about the male reproductive system. We're not 100% through this yet. So we still have to talk about the testosterone hormone itself. We do still have to talk about the male sex act and how it proceeds. But I feel like I've given you almost an hour and a half of content at this point, so I feel like maybe this is a good time to cut this short. So let's leave our lesson off here. Next time we will pick up with finishing the male reproductive system, talk about male sex hormones and the male sex act, and then we will move on to the female reproductive system, which I suspect is probably going to take us more than one lesson to complete. All right. Thank you for your attention. Hope you have a good rest of the day. We'll talk to you soon.